Welcome to our senior secondary biology. In this video, we're going to take a look at cells. Let's get started. So what are the learning outcomes? At the end of this video, I expect you to know what a microscope is. You should also be able to identify its parts and their functions. I expect you to be able to calculate magnification. I expect you to describe a typical cell. Finally, I expect you to distinguish between a plant and an animal cell. This is where this video is going to end. In the next video, we are going to attain or try to attain these outcomes. Describe different types of cells. So a cell is a basic functional block of a living thing or of an organism. So the keywords here are basic, which means the simplest possible; functional, meaning something that's working, that has got a function, that does something; block are smaller units that makes up something big. So that's what a cell is. Let's say I've got this living organism here. So if we get or we take a snapshot of a smaller part of it, let's say this part, there are smaller blocks that makes up this living organism. So that's these small units, small blocks, are what we call in biology as cells. So we're going to study cells because a living organism must have cells. So for us to study cells, we're going to start with an instrument that is used to study cells, or that is used to view cells. Cells are very small, such that we cannot see them with our naked eyes. So we have to make use of what is called a microscope, which enlarges or magnifies smaller objects. Here is an example of a microscope. By the way, a microscope is used to see anything that's small. It's not just cells, even bacteria. Normally when we're eating food or when we are doing anything, even when we are exposed to dirt, we don't see the bacteria that we're exposed to, and we can only view them using a microscope. So when you want to start something using a microscope, what you do is you get whatever you want to study, which in science, anything that you are studying is called a specimen. So you can put it here on this part here, which is called a stage. So once you put it on the stage here, you can choose one of these lenses called objective lenses. So these have got some glasses attached to them here. These glasses have got what is called magnifying power. They have got different magnifying power. Magnifying power is somehow related to the level of details that you can view or that you can see about a specimen. In short, this lens here can give you more details as compared to this lens or this lens. So these have got different levels of details depending on what you are interested in. So once you put your specimen here on the stage and you choose one of these objective lenses, you look through here. So this is called ocular lens or eyepiece. So through the eyepiece, you look through here and you'll be able to see the object that's been studied. Another part of the microscope is here called the mirror. So what the mirror does is, if there is light from an external source, that light will come and reflect here. So it will come here, then it will be reflected. There is an opening here, so it will be reflected and it will go to where the object is. Note that this opening, this is one end of the opening, and here is another end of the opening. So once you place the object or you mount your object there, the light from the mirror will pass through here and you'll be able to see your object clearer. So the function of the mirror is to provide illumination or lighting to the object. Other parts of the microscope that are of interest are Of course, this here, which is called the base, which just supports the entire structure of the microscope. And we also have got these, which are called stage adjustment knobs. So these are like, you can turn them, they're like screws. So with this, you can adjust the stage. You can tilt it to this side or this side, depending on how you want to position it. We also have got these two knobs here, which are called adjustment knobs. This is stage adjustment knobs, and these are just adjustment knobs. There are two. One is called fine adjustment, and the other one is called coarse adjustment. These are used to adjust the lenses here. So depending on maybe you would want to change the position of the lens, you want it to be somewhere here, so you can just use these to turn this, which is called the head. So you can turn the head so that the lenses are positioned the way you want. So let us label these various parts of the microscope. So this is the arm, which is used just to hold the upper parts of the microscope, or to carry the microscope. So if you want to carry the microscope, you want to lift it, you use the arm. We also have got the ocular lens or eyepiece. We have the objective lens or objective lenses, as they are called collectively. We also have got the stage, the mirror, the base, and stage adjustable knobs or stage adjustment knobs, and the adjustable knobs, these two here. Of course, I've just shown the arrow that shows one, but even this one is part of the adjustable knobs. So just a quick summary of the functions of the various parts of the microscope. So I've got the base, which provides support to all other parts of the microscope, the arm, which gives support to the head of the microscope and used to carry the microscope, the eyepiece, which is used for looking through the microscope. Also, I've got the objective lenses, which are used to magnify the object. So the objective lenses are the ones that do the actual magnification because of the lenses or glasses that are attached to them. We also have got the stage adjustable knobs or adjustment knobs, which are used to adjust the stage of the microscope for proper focusing. We have those two adjustment knobs, which are used for adjusting the lenses of the microscope. Also, I've got the stage where the specimen is placed for viewing, and the mirror, which is used to provide light to the specimen being viewed by reflecting light from an external source to it. The last part when it comes to studying the microscope is magnification. So I'd shown that magnifying is just the same as enlarge. So you can say to make magnification means to make bigger or enlarge. The magnification of an object is how much an object has been enlarged. So remember, the whole purpose of using a microscope is to make something that's small look bigger. And we have to study this magnification because it gives us information about how big the image or what you are seeing on the eyepiece is as compared to the actual image. I mean to the actual object or to the actual specimen. The formula for magnification is size of image or drawing over size of object or specimen. So when you are looking through the eyepiece, it can happen that you want to draw what you are seeing. So what you see through the ocular lens has to be translated or has to be drawn on a piece of paper, so that you've got an idea or so that you've got a record of the details that you are seeing. So for you to get magnification of what you have just drawn, what you have drawn is the image or drawing. It's not the actual thing. It's not the actual size of the thing. So you divide this by the actual size of the object or specimen. That's how you get magnification. Magnification has no units. So if, for example, whatever I've drawn is 2 centimeters or 2 millimeters, doesn't mean that when calculating magnification, whatever number you're going to get it to have millimeters or centimeters at the end. We represent or we write magnification like this, by 4 or 4 by. So if the number you get here is 4, so you write times 4 or 4 times, mean that your object has been enlarged times 4 or 4 times or by 4 or 4 by whatever you may call it. So this is how we calculate magnification and how we represent magnification. So here is an example. The question says a student drew a specimen to be 20 millimeters when it is actually 10 millimeters. What is the magnification? So the drawing was 20 millimeters, but the actual specimen or object is 10 millimeters. How do we calculate magnification? So we'll call the formula magnification is equal to size of image or drawing over size of object or specimen. What is the size of image or drawing in this case? So the student had drawn 20 millimeters, so the size of the drawing is 20 millimeters, and the actual size of the object or specimen, like I said before, is 10 millimeters. So we can use these two values. So we're going to have magnification equal to 20 millimeters over 10 millimeters. When we divide, we'll get 2. 20 millimeters divided by 10 millimeters is 2. And our magnification will be by 2 or 2 by. Now that we have studied the instrument that is used to study cells, we can now start our studying of cells. So we've got different living organisms. We've got plants, we've got animals, we've got bacteria, and each class of living organisms has got the same type of cells. In short, an animal and the plant have got cells that look different. In this case, we're going to start with a simple structure of an animal and plant cell. So we're going to start this, and later we're going to look at the details structures of the animal and plant cell. So an animal cell is this one. So all animals have got cells that look like this one. Well, all plants have got cells that look like this one. The two cells share they've got some similarities and they've got some differences. One of the common things they share is this black-like thing here, which is called a nucleus. By the way, these structures that makes up a given cell are called organelles. Other than the nucleus, the two share this fluid-like where the nucleus is in this case, in the plant cell, it is this here. So this fluid-like where the other organelles are floating is called a cytoplasm. Finally, they share this boundary here, which is like a covering for the entire cell. And for the plant cell, it has got two borders, two coverings. The first one is this one, the second one is this one. So this, the one that is inside, is the one that is that in the animal cell is this one. In short, it is made of similar things, and it is called a cell membrane. So these are the organelles or the features that the two share. What about the differences? First and foremost, the most common difference that one should recall is the way they look. So you can see that an animal cell has got an irregular shape, or a shape that is not regular, in that you cannot measure it using a meter rule. Well, a plant cell has got a regular shape, which means you can use a meter rule to measure its dimensions. So that's one of the observable differences. Another difference is that a plant cell has got this organelle called cell wall. So an animal cell doesn't have this. Also, a plant cell has got these organelles here. So this, and this. So I've colored this one green so that you can remember it, and you can remember its purpose. So this is what makes most trees and plants green. Animal cells don't have this. These other ones are not in green, just because I want you not to get confused when you see something like this. Your exam papers are usually in black and white, so you see this, not this. So this is called a chloroplast. A chloroplast. Other than this, a plant cell has got these large central permanent vacuole. That's what it's called. An animal cell doesn't have this. So these are some of the differences. Let's tabulate these differences so that we've got a reference. So these are the differences. The first one is on the left, by the way, it's plant cells. On the right, animal cells. So the one that we have discussed is the cell wall. So plant cells have got a cellulose cell wall. So what this cellulose means is this structure is made of a compound called cellulose. Well, an animal cell doesn't have this. Also, a plant cell has got a large permanent vacuole. Well, an animal cell does not have a large permanent vacuole. There are times when you're viewing an animal cell under a microscope, you can see something like a vacuole, but whatever you see, it won't be permanent. Well, plant cells have got permanent vacuole. A plant cell also has a chloroplast. Well, an animal cell does not have a chloroplast. Otherwise, if an animal cell had a chloroplast, we could be appearing green like the Hulk. Also, a plant cell has got regular shape and size, while an animal cell does not have a regular shape and size. So these are the differences between plant and animal cells. So the main parts of any cell are the cell membrane, the nucleus, and the cytoplasm. Collectively or together, they are called protoplasm. So let's take a look at a detailed structure of a cell. We're going to start with an animal cell and we'll move to a plant cell. So these are the organelles that makes up an animal cell. We're going to discuss most of them here. Maybe the ones that won't discuss are not that relevant, but we'll discuss most of them here and their functions. We'll start with a cell membrane. So a cell membrane is a sheath or covering around the cell, and it acts like a gate in that it allows certain substances to pass through it. Because of that, it is termed to be partially permeable or selective permeable. What this means is it permits only certain substances to pass through it, and therefore its function is just to allow certain substances to enter or leave the cell. Another organelle is the nucleus. So this is how the nucleus looks under a microscope that gives more details. It has got other organelles inside it, such as the nucleolus and these threads here. The function of the nucleus is to control all cellular activities. Another organelle is the vacuole. So we discussed that animal cells don't have vacuole, but some do have vacuoles, and those vacuoles are never permanent and they are very small. So what I mean that they're never permanent is if you view a cell, you can see the vacuole, and at some other point, if you view the same cell, you might not see the vacuole. So the function of the vacuole, even though it's small, it's the same as the function of the vacuole in plant cells, where it is big, and it is to house substances like food and water for the cell. So they act like storage. So whenever there's something that has to be stored, it is stored in the vacuole because it is the storage for the cell. Also, I've got these ones which are called mitochondrion. So this is the mitochondrion, and together they are called mitochondria. So the function of the mitochondrion is to act as the powerhouse of the cell. If a cell can be viewed to be a country like Zambia, then a mitochondria is responsible for manufacturing energy or power. So this is done through the process of respiration. So the manufacture of energy is done through the process of respiration, which we discussed to be one of the characteristics of living things. And respiration is where energy is made using food and gases. We discussed this when we were looking at the characteristics of living things. Another organelle has the vesicles. So the vesicles are these small organelles here. So these are transport agent and they are always going towards the cell membrane. So we say they are membrane bound and associated with transport. How do they do that? So some organelles here, such as this and this, well, we haven't discussed them yet, but we will. They manufacture substances like proteins, fats, and the likes. Once they do that, there's another organelle here, the Golgi apparatus, which we're going to discuss its actual function, which packages the manufactured substances into the vesicles. The vesicles now carry the manufactured substances to the cell membrane where they transport outside the cell. So they're always heading towards the membrane. That's what it means by membrane bound. And of course, they are transport agents. Another organelle is the endoplasmic reticulum. Endoplasmic reticulum. So these are the endoplasmic reticulums. In short, we call them ER. So ER for endoplasmic reticulum. So there are two types of ERs. There is SER, which is smooth endoplasmic reticulum, and RER, which is rough endoplasmic reticulum. So the smooth endoplasmic reticulum doesn't have what are called ribosomes. Well, rough endoplasmic reticulum has got ribosomes. We're going to discuss what are ribosomes and their functions. So what's the function of this smooth endoplasmic reticulum which doesn't have ribosomes? So the function of this is to manufacture fats and steroids, and it also contains enzymes that detoxify toxics. So it has enzymes. Enzymes are biological catalysts. In short, enzymes are substances that speed up a reaction without them participating or being changed in the reaction. So they have got enzymes that somehow help to make toxic or poisonous substances not harmful. The other one is the rough endoplasmic reticulum. So this rough endoplasmic reticulum has got ribosomes. And ribosomes, actually what they do is they manufacture proteins. And since they are here, so this is where proteins are manufactured. And like I said, ribosomes, they are in two types. There are free ribosomes and those which are attached to the RER. So the free ribosomes are these here, and the ones that are attached to the endoplasmic reticulum are here. And of course, the function is the same, which is to manufacture proteins. We also have the Golgi apparatus. I don't know if it's Golgi, others call it Golgi apparatus. But what the Golgi apparatus do is, once proteins, fats, and steroids are manufactured in the endoplasmic reticulum, this is where they are stored, modified, and packaged into vesicles for them to be transported. We have the cytoplasm. So the cytoplasm is the fluid where all the organelles are. So in short, this is where all activities in the cell take place. So I'm going to finish this video by studying a plant cell. Most of the organelles here, we've already discussed them in an animal cell, and they have the same functions. There are some organelles which are only found in a plant cell, such as the chloroplast. So we're going to concentrate on these new other organelles. I'll start with the vacuole. So you can see that in a plant cell, we have got a large central and permanent vacuole. So unlike in an animal cell where we might see vacuoles at times, and at times we cannot see them and they are small and there might be more than one, in a plant cell we've got one large permanent central vacuole. The function, of course, is the same as in an animal cell. Another organelle is the chloroplast. So in a chloroplast, it's where photosynthesis takes place. So photosynthesis is the process by which food is made using sunlight energy, and it happens inside the chloroplast. Another organelle is the cell wall. In an animal cell, we discussed the cell membrane, which we say to be partially permeable. The other word for partially permeable is selective permeable, which means it allows certain substance to pass through it. Unlike the cell membrane, the cell wall is said to be fully permeable, which means it allows any substance to pass through it. Its function is not to allow what goes through it or not, but to protect the cell and give the cell its shape. So this marks the end of this video on cells. The next video will concentrate on cell specialization. Make sure you like, subscribe, and share. Of course, if you're using Facebook or YouTube, you can use the comment section to comment any detail that you feel you need clarification. Otherwise, happy solving.